All right, ladies and gentlemen, welcome back. So today we're going to really get into the actual processes of um, this cellular respiration. Before I really jump into this, though, let me just back you up and say that we could go into excruciating detail on every chemical reaction that happens in every process of cellular respiration. I have no intention of doing that. If you take a biochemistry course in college, that may be something that you are required to memorize at that point. What I want you to get at here is the bigger picture. What are these processes doing? What is their overall function? What are we putting in as major inputs? What's coming out as major outputs? And why do those inputs and outputs make connections from one step to the next? So, with that being said, let's start with glycolysis, right? And so when we break that term glycolysis down, we are literally saying we are splitting or breaking sugar. So since we are tracing this process with respect to glycolysis, we're going to take that 6-carbon glucose molecule and break it down into 2 pyruvates. Each of those pyruvates contains 3 of those carbons. Glycolysis is going to be a process that happens in your cytoplasm. That's going to become important later when we talk about some of the evolution of how this process probably came about. It is also anaerobic. There is no requirement for oxygen here. When we look at glycolysis, there are a handful of chemical reactions. I'm going to take those chemical reactions and lump them into 2 pieces. What we're going to talk about as your energy investment phase versus your energy payoff phase. And if you look at what those terms are and look at what's going on, we're really talking about, am I having to input ATP to get the process to go, or am I getting excess ATP out? So when I look at that initial set of chemical reactions to take that glucose and start breaking it down, it is a chemical reaction. It is breaking things down. It is going to require some energy to do that breaking. And so we're going to take that glucose and invest 2 ATP molecules to break it in half. There's a bunch of enzymes in here that are helping to catalyze that, lower the amount of energy required, but I still need some energy to go in. The energy payoff phase on the other side gets me some energy back out. Ultimately, this is an exergonic reaction or an exergonic process, exergonic pathway. And so we're going to use some of that redox and some of that energy and be able to get 4 ATPs back out in that energy payoff set of chemical reactions. That's also where we're going to shuffle some electrons and some hydrogens around to create some NADH that is ultimately going to go to your electron transport chain. We get those 2 pyruvate molecules out at the end. We also net a little bit of water here. So input is that glucose, ADP and some phosphates, some NAD plus, electrons and hydrogens. We ultimately get those pyruvates out, some water out, some ATP out, NADH and some hydrogen ions as well. All right. So we ultimately gross 4 ATPs. We get 4 ATPs out of this overall process. But if you look at this, we're really only going to end up netting 2 ATPs that cell can go use somewhere else, because 2 of those ATPs that we get here are going to get fed back in that energy investment phase to keep this process moving. All right, so at the conclusion of glycolysis, we have produced those pyruvate molecules, and here is what one of those molecules looks like. Here's the thing, in order to get that pyruvate into the next major phase of cellular respiration, which is your citric acid cycle, we have to move those pyruvate molecules from the cytoplasm into the mitochondrial matrix. And in order for that to happen, that means it has to pass through those mitochondrial membranes, both its outer and inner membrane. And it's going to end up happening via a transport protein. It's actually active transport. We need to be able to pump up the concentration of this pyruvate in order to keep that citric acid cycle going. But in order to get that pyruvate into the mitochondrial matrix and then ultimately be in a form that is capable of being taken into that citric acid cycle, we have to kind of do this intermediary step called grooming. And so in the presence of oxygen, that pyruvate will enter the mitochondria and it will go through this grooming process in order to form this molecule called acetyl-CoA. And that coenzyme is really going to be the important part that we attach to that pyruvate so that it can be entered into that citric acid cycle and we can do that process. So what we're looking at here is here's a single pyruvate molecule. It's going to go through that transport protein, and in that process, we're going to pull off carbon dioxide. And so remember that we're taking a 6-carbon glucose, breaking it down into 2 pyruvate molecules, each with 3 carbons apiece. So here's 3 of those carbons that came from that glucose. Well, one of those carbons plus 2 oxygens are going to come out as carbon dioxide. We then breathe that out as a byproduct. In the process, now I've created a blank bond here that I can attach that coenzyme A to. There's a couple other intermediary chemical reactions that are happening here that I'm not showing you, but in the process, we also do a little bit of redox going on and create NAD plus into NADH, as well as some extra hydrogens from, again, things that we're just not showing that are happening here as intermediaries. So what do you need to know? Well, the pyruvate goes in, NAD plus goes in, the coenzyme A goes in. Those are all our major reactants for this grooming process. Coming out of that, we yank this carbon and oxygen off to get carbon dioxide, which will get released as a waste product. We'll shuffle some electrons and some hydrogens around to get NADH, which will get shuttled over to the electron transport chain we'll talk about a little later. And we're also able to then attach that coenzyme A, which is what you're seeing here, to make this molecule called acetyl-CoA. Now keep in mind, this diagram is showing you a single pyruvate. So if we were to trace what's happening for a single glucose molecule, which is often what AP likes to ask you about, remember that that means this grooming process has to happen twice. So 2 pyruvates go in, 2 carbon dioxides come off, 2 coenzyme A's go in, 2 acetyl-CoA's come out, 2 NAD pluses go in, 2 NADH's come out. So just keep that in mind. We've got to double everything because a lot of the diagrams are showing you for a single pyruvate, and yet we're feeding in a glucose at the beginning of glycolysis. All right, so once we have moved through this grooming step, which is what you're seeing here, that acetyl-CoA is then going to get fed into our citric acid cycle. Remember I said you can also come across the citric acid cycle called the Krebs cycle. So whether you see Krebs cycle or citric acid cycle, it's the same process. It's the second major component to cellular respiration, where we're going to finish the oxidation of the carbons from glucose. We're going to really finish the breakdown here. So we're going to take those pyruvates, turn them into acetyl-CoAs, feed them into this cycle. So when we look at major reactants, your acetyl-CoA is going to come in here. We're also going to make some more electron carriers, which is going to be the big function for the citric acid cycle. So we're going to make a handful of NADHs, which means I need NAD plus to start. I'm also going to make another electron carrier we haven't come across yet, FADH2, so we're going to start with FAD. And we're also going to make a little ATP out of here, so we'll have to start with some NAD plus, or NADP, I should say, plus phosphates. Products, that coenzyme is going to come back out. That's a big component to this cycle, is that we're going to take these carbons in here, be able to shuffle them around, move electrons around to create these electron carriers and the ATP. But it's a cycle. And so I'm going to be able to get that coenzyme back so that it can get fed back into this grooming component. I will also get some carbon dioxide out of here, so that will come off as a waste product. And again, I said we'd make some NADH, ATP, and FADH2. Now again, remember that this is showing you a single pyruvate. So if you're asked about, well, what does the citric acid cycle make per glucose, you've got to double all these numbers. All right, so it'll be 4 carbon dioxides, 6 NADHs, 2 ATPs, and 2 FADH2s. So this shows the full process, all of the intermediary molecules that get made, where things come in and come out. Um, you can see we use some coenzymes here again as well, where the ATP comes out. Um, each of these steps really does ultimately be able to explain that full redox process with the reducing agents, the oxidizing agents, where all the products gets made. Here's that citrate, right? That's the first product that gets produced, which is where the citric acid cycle term comes from. As far as I'm concerned, you can dig into this as deep as you want, but I really need you, for our purposes this year, to be able to know this component. Um, Other than maybe saying you probably should know that citrate exists here as that first product, I don't need you to memorize all of these components. That being said, you get to a college course, biochemistry course, and this may be something that you need to memorize. So the more that you can pick up now, perhaps the better off you are in the future, but I don't need you to know all of this for us right now. That all being said, you should be able to realize that each step is catalyzed by its own enzyme. So enzymes are going to be a huge important component of this cycle. Um, and that, again, that citrate probably is a good thing to know that that is, in fact, the first um, molecule that gets produced, because that's where the name of the cycle comes from. The rest of those steps are really about that decomposition of citrate back into this molecule called oxaloacetate that keeps this process going. So we're going to add this, move that coenzyme A off to attach these carbons from the glucose, and that's what keeps this cycle moving and allows us to produce all of these electron carriers as well as a little bit of that ATP.